Welcome all to AK Bio lecture series. At the end of this class, you will understand how the egg is protected from the multiple sperm entry, means how the process of prevention of polyspermy occurs. So let's start. During sexual reproduction, a zygote nucleus is formed by the fusion of sperm and egg nucleus to produce new diploid organism. In most animals, only one sperm fuses with the egg during normal fertilization, and this is known as monospermy. So here you can see the difference between the monospermy and polyspermy. I am not describing this table. You can go through these details by pausing this video. However, huge number of sperm usually reaches the egg to ensure the fertilization of all spawned eggs during external fertilization, and this produces the risk of polyspermy. More than one sperm entering the egg during fertilization. So here you can see the cartoon representation of this competition of sperms to fuse with the egg. So as polyspermic eggs typically undergo abnormal cleavage and die shortly after fertilization, animals have evolved several elaborate mechanisms to exclude excess sperm during fertilization. That mechanism is called blocks to polyspermy. First of all, the animals adequately regulate the number of sperm simultaneously reaching the egg. In external fertilization of monospermic frogs, for example, insemination with relatively high concentration of sperm suspension, that means at least 10 raised to 5 cells per ml, is necessary to achieve fertilization. So, only in the presence of this much huge quantity of sperms, the fertilization is possible to the monospermic frog species. So in this case, more than 1000 sperms can reach the outside of the extracellular cords. That means the jelly layer, white line envelope surrounding the egg membrane. Due to this huge number of sperms, it is obvious that the more than 1000 sperms will reach this egg membrane. That means outer layers of the membrane. However, it is estimated that approximately 20 sperms reach the egg membrane. Even though the more than 1000 sperms can reach outside the extracellular cords, jelly layer, and even to the vital envelope, only 20 sperms can reach the egg membrane, that is plasma membrane, at an interval of 2 to 20 per second over 20 minutes. So based on these two facts, that is two interesting facts of the number of sperms that reaches the egg cell membrane, means only the 20 sperms in the period of 2 to 20 seconds over 20 minutes, we can conclude that the extracellular cords of eggs, that is this egg jelly and the vital envelopes or the other extracellular cords, that prevents or play an important role in reducing the number of effective sperms that and delaying the arrival of other sperms following fertilization by the primary sperm. So the extracellular cords of the egg that play a crucial role in the prevention of polyspermy by reducing the number of sperms that reaches the plasma membrane of the egg. And we are discussing about the types of prevention of polyspermy, and the two most widely studied are called the fast block and slow block to polyspermy. As their names imply, they block these blocks occurs at different times to fertilization. One is very fast and one is very slow, comparatively slow after fertilization. So the fast block, first one is we are discussing the fast block. The fast block to polyspermy is mainly used by eggs from external fertilizers. So fast block is exclusive for the external fertilizing organisms. In the case of internal fertilizing organisms, especially in the case of mammals, the fast block is not reported. Only slow block is present in the case of mammals and internally fertilizing organisms. And slow block is used by eggs from nearly all sexually reproducing organisms. So slow block is common to the external fertilizing organisms and internal fertilizing organisms. And the slow block to polyspermy, eggs release material into the extracellular space, that is perivitelline space, and within minutes after the fusion of egg sperm plasma membrane and transforms the egg to a structure unable to permit fertilization. Means in the process of slow block to polyspermy, the eggs release certain chemicals into the perivitelline space and makes the membrane rigid and more strong to prevent the entry of further sperms into the egg plasma membrane or egg cytoplasm. So the initial, of course, the first reaction is fast reaction. So this is the initial reaction accomplished by the electrical change in the egg cell membrane, and the slower reaction is caused by the exocytosis of cortical granules. I already mentioned that some chemicals are released from the egg cell that is from the cortical granules. We already discussed about the cortical granules which are present in the cortex region of the egg cytoplasm. So only the cortical granules the chemicals from the cortical granules are released into the perivitelline space and makes the membrane more rigid. So let's discuss the first block to polyspermy in detail. And this block was subsequently identified in many monospermic animals, including both vertebrates and invertebrates, by intensive investigations for 4 decades. And although fertilization signaled depolarization, means in this fast block polyspermy, we already mentioned that the fast reaction is accomplished by the electrical changes in egg cell membrane, and the signaling pathway that activate the fast block, including the molecular identity of the channel that conducts the depolarization currents, was not yet known for any other species. So in detail how the mechanism of activation of the depolarization is not well known in the case of other animals, but in the case of sea urchin and is well established. So fast block to polyspermy, once one sperm has fused with the egg, that is the monospermic species, that is peculiar feature of monospermic species, and once the sperm has fused with the egg and a soluble material from the sperm, that is NADP, enters into the egg cytoplasm. Here you can see the entry of the sperm in different stages. We can number this as 1, 2, 3, 4 stages, and in the first stage it is very near to the egg jelly layer. In the case of second stage, it starts to cross this jelly layer, and in the third stage the chromosome process formation has completed, and then the sperm reaches to the plasma membrane of the egg, isn't it? So once one sperm has fused with the egg, the soluble material from the sperm, that is NADP, so not NADP, NAADP, AA means nicotinic acid adenine dinucleotide phosphate. Okay, so NADP that enters into the egg cytoplasm, and within 1 to 3 seconds after the binding of the first sperm, NADP. Here you can see the NADP. Consider this is egg, and this is the cytosol of the egg. The upper portion is the cytosol of the egg, and the down region, that is which is the region of endoplasmic reticulum or endoplasmic lumen, and here you can see once the sperm has fused with the egg plasma membrane, the cytoplasm, the NADP from the sperm enters into the cytosol of the egg. So NADP, which is bounded to the TPC, that is a transporter or the channel, and that means NADP sensitive calcium and sodium channels, that is TPC, and on this is present, that is TPC is present on the endosome membrane, and the NADP is bounded to the TPC with a high specificity and nanomolar affinity. And the membrane potential shifts to a positive level as a result of the efflux of calcium ions Here you can see once the NADP bounded to the TPC transporter or TPC channel or on the endosome membrane, then the TPC gets opened and the TPC transport or the TPC transport the calcium or Na plus sodium ions from the endosome into the cytosol. So inside the cytosol, the positively charged calcium and sodium becomes in high concentration when compared to the outside of the cell, and it becomes it results in the change in the membrane potential also. So the membrane potential shifts to a positive level as a result of the positively charged ions, that is calcium and sodium richness inside the cytosol, and which reaches about a plus 20 millivolt with respect to the exterior of the cell, and is the result of small influx of sodium into the egg through newly opened sodium channels and opened TPCs. At the same time, some sodium channels on the plasma membrane of the egg is also gets opened with the unknown mechanism, and the endosome membrane on the endosome membrane the TPC is also opened. From endosome the calcium ions and the sodium ions is transported outside the endosome towards the cytosol, and the sodium channels that open on the plasma membrane that transport the sodium ions from the outside of the cell towards the egg cytoplasm. That results in the small influx of sodium into the egg that results in the change in the membrane potential towards a shift to the plus 20 millivolt level. So that is called that results in the change in the potential, and the fast block to polyspermy is activated by changing electrical potential of the egg cell membrane that occurs immediately upon the entry of sperm. But the process of the activation, the change in the membrane potential is very quick in action, and this occurs immediately upon the entry of sperm. And after this event, extra sperms cannot fuse with the egg cell membrane that have positive resting potential. Because of the positive resting potential, that is plus 20 millivolt of the egg cell membrane, the further sperms cannot enter into this fertilized egg, so-called fertilized egg. So the shift means the no more sperms can fuse to the egg. Okay, but it is not a permanent way of prevention of polyspermy. This is the first response of the egg that senses the entry of one sperm into the egg cytoplasm. Okay, since it is the fast block is a transient and it is not a permanent way of prevention of polyspermy, since the membrane potential of the Sea Urchin egg remains positive for only about a minute. So because this positive charge plus 20 millivolt is not a permanent charge of this the plasma membrane, it will shift to the normal resting membrane potential within minutes. So the cell have to evolve another mechanisms to prevent the polyspermy, that is the slow block to polyspermy. However, given that the fertilization potential is a transient, this temporary block is followed by a permanent block, that is slow block. Only this slow block is the permanent way of prevention of polyspermy, and fast block is the first quick reaction of the egg to prevent the polyspermy. This brief potential shift is not sufficient to prevent polyspermy permanently. Means the fast block is not sufficient to prevent the polyspermy permanently, and the polyspermy can still occur if the sperm bound to the vitelline envelope are not somehow removed. So we know that more than one sperms or multiple sperms are already attached to the vitelline envelope of the egg. Even one sperm enters into the egg cytoplasm, and they release NADP. Okay. NADP. So this the egg has to remove these extra sperms from the vitelline envelope, and this is accomplished by the cortical granule reaction, that is the slow block to polyspermy, also known as the slow block to polyspermy. Okay. Then cortical granule reaction is also known as slow block to polyspermy, and this becomes active about a minute after the first successful sperm fusion. So this mechanism of slow block to polyspermy has initiated within minutes, not within seconds. Within seconds, the fast block is will happen, but the slow block to polyspermy is a slow process, so it becomes active only after a few minutes, first few minutes. Okay. Of the sperm egg fusion. And it is found in many animals, animal species, including sea urchin, and most mammals, including human. So this is the whole process of slow block to polyspermy, and we will discuss this in coming slides. So we can divide the whole process of slow block to polyspermy into two: the activation prior to gamete and activation after gamete fusion. So what is activation prior to gamete fusion? In sea urchin, the binding proteins on the sperm acrosomal process, we already discussed about the binding proteins and its function, and the binding proteins of the sperm binds to the binding receptors on the egg cell surface, and receptor activates a protein called tyrosine kinase or SRC kinase, which activates phospholipase C, PLC. Here you can see the activation after gamete membrane fusion. Here you can see the activation prior to gamete fusion. Here you can see the binding proteins get attached to the sperm receptor, and the receptor activates the another protein called tyrosine kinase, which is a SRC family protein, and this SRC family protein, tyrosine kinase, activates another protein called PLC. And the PLC cleaves the PIP2 into two molecules, that is PIP2 into DAG and IP3. And the DAG is then continue with the by attaching to the plasma membrane, that is inner leaflet of the plasma membrane, but IP3 is fall into the cytoplasm of the egg, and this IP3 is freely present inside the cytoplasm, and the IP3 gets attached to the ER membrane receptors, that is calcium channels, and the IP3 opens the calcium channels of the endoplasmic reticulum, and the endoplasmic reticulum is the sink of calcium ions, and the endoplasmic reticulum is the calcium depository area of the cell, and the endoplasm, once the calcium channels gets opened on the endoplasmic reticulum, the calcium efflux from the endoplasmic reticulum into the cytoplasm occurs. And this is all about the activation prior to gamete fusion by binding protein with the sperm receptor. Okay, then activation after gamete fusion. Once the sperm has fused with the egg, the soluble material from the sperm, that is NADP, enters into the egg cytoplasm. We already discussed that, and NADP brought in by the sperm, that is activates the G protein and SRC protein. Yes, here you can see the entry of the NADP. So once the NADP brought into the sperm, by the sperm activates the G protein, that is SRC activation, and activates SRC activates enzyme phospholipase C, and phospholipase C cleaves the membrane phospholipid, that is PIP2 into IP3 and DAG. Here you can see the PIP2, and PIP2 is cleaved into DAG and IP3. And IP3 binds to IP3 receptor in these calcium channels of the endoplasmic reticulum. As a result, release calcium into the cytoplasm by opening these calcium channels. So this IP3 bound to the calcium channels on the endoplasmic reticulum, and the cytoplasm and the calcium enter into the cytoplasm by opening the calcium channels. Throughout the animal kingdom, it has been found that IP3 is the primary agent for releasing calcium from the intracellular storage, such as the endoplasmic reticulum. So, in different cell signaling system, you can see this action of IP3 to activate the calcium channels on the endoplasmic reticulum, and as a result, the calcium gets released into the cytosol from the endoplasmic reticulum, which is a common pathway in different signaling pathway. Okay. So the way calcium is released varies from species to species, and sea urchin and mammals, the rise of calcium concentration responsible for the cortical granule reaction is stored in the endoplasmic reticulum of the egg. So here you can see the another diagram that represents the slow block. Here you can see the action of phospholipase C, that is phospholipase C cleaves the PIP2, and PIP2, as a result of the cleaving of PIP2, the one portion of the PIP2, that is DAG, remains attached to the plasma membrane, that is inner leaflet of the plasma membrane, and the PIP2 gets released into the cytoplasm, and the IP3 starts its IP3 pathway, that is IP3 binds to the receptor on the endoplasmic reticulum, that is calcium receptor, and it is also a calcium channel also. So calcium channel gets opened by IP3 binding, and the calcium is released into the cytoplasm from the endoplasmic reticulum of the egg, activated egg. So the calcium level inside the cytoplasm gets increased. Okay, as a result, as soon as calcium is released from the endoplasmic reticulum, the cortical granules fuses with the cell membrane above them. So here you can see it is not represented in, it is not mentioned in this diagram. Here you can see in the cortex region the vacuoles or granules. Inside the granules, lot of chemicals are there. So these granules gets attached to the plasma membrane and releases contents to outside. To outside means to the perivitellin space. Okay. So the cortical granules are themselves tethered to the cell membrane by a series of integral membrane proteins, that is snare. And I mentioned that there will be a lot of cortical granules. Okay, so the cortical granules are themselves tethered or already attached to some proteins called snare proteins that facilitate calcium mediated exocytosis. So the snare proteins you will see the snare proteins role in the fusion of the plasma membrane of egg and the sperm in coming slides. So keep in mind that snare proteins is involved here also. That is, they are already attached to the cortical granules. So the cortical granules exocytosis is also facilitated by snare proteins. And DAG and calcium, the same snare protein is functioning in the fusion of plasma membrane of the egg and sperm. Okay. And DAG and calcium from the ER, that is endoplasmic reticulum, activate another protein called PKC, protein kinase C. This DAG activates a lot of proteins, that is including the protein kinase C, which in turn activate another channels, that is sodium hydrogen ion exchange pump. So DAG activates PKC, and the PKC activates this channel, this pump, sodium hydrogen ion exchange pump. So this is an antiporter. Okay, antiporter. Once released calcium can diffuse directly, or it can facilitate the release of more calcium by binding to the calcium triggered calcium release receptors, also located on the cortical endoplasmic reticulum. We already mentioned that the presence of cortical granules. So here you can see in this diagram, you can see this presence of cortical granules directly beneath the Sea Urchin egg cell membrane are about 15,000 cortical granules in the cortex region of the cytoplasm, and each cortical granules is in the diameter of 1 micrometer, and upon sperm entry, cortical granules fuses with the egg cell membrane by this mechanism, based on as a result of the signaling mechanism as described in these slides, in these previous slides. So upon sperm entry, cortical granules fuse with the egg cell membrane and release their contents into the space between the cell membrane and fibrous mat of vitellin envelope. So that space is called the perivitellin space. Okay, and the components of the cortical granules bind to the vitellin envelope form fertilization envelope. So because of the presence of because of the release of the chemicals from the cortical granules into the perivitellin space, the perivitellin space get the space between the plasma membrane and vitellin membrane transformed into another envelope, that is called fertilization envelope. So after the releasing of the cortical granules, the space is called fertilization envelope. The fertilization envelope starts to form at the cytosperm entry and continues its expansion around the egg. Consider this, this is the egg plasma membrane, and this is the vitellin envelope, and the sperm has entered into this area, and the cortical granules, these cortical granules are 15,000 cortical granules are present on the cortex region also. So once the sperm has reached into the plasma membrane or into the vitellin space, the cell signaling occurs inside the cytoplasm, as we described in the previous session. Okay, then once calcium level has increased, inside the cytoplasm, the cortical granules get attached to the plasma membrane of the egg and release its contents to the perivitellin space and transform the perivitellin space into fertilization envelope. So, but the process of fertilization envelope formation started at the point of sperm entry, and then it will expand to the other parts also. Okay, and this process starts about 20 seconds after sperm attachment and is complete by the end of first minute of fertilization. And several proteins are released by cortical granules exocytosis. Which are the proteins that are present in the cortical granules are serine proteases, mucopolysaccharides, peroxidase, and hyalin. These four proteins all have the different functions in the process of slow block to polyspermy. I think you are familiar with this picture if you followed the last video of this channel, and in that video we mentioned about this microvilli, what is microvilli, and I think you are also familiar with this picture also. Yes, this is the plasma membrane and this is the vitellin envelope, and these connections between the plasma membrane and vitellin envelope are called posts, protein post. So serine proteases that cleaves the protein post that connect the vitellin envelope proteins to the egg cell membrane. And it also clip off the binding receptors and any sperm attached to them. That means if any sperms get attached to the vitellin envelope, that connections will be removed by serine proteases. And mucopolysaccharides are viscous compounds that absorb water to expand the space between the cell membrane and the fertilization envelope. Here you can see the expansion of this area. Initially, it was very small area like this one, isn't it? After release of the cortical granules, here you can see in this diagram, second diagram, you can see here after release of the cortical granule of the chemicals from the cortical granules, this area becomes expanded. Okay, so after few seconds, you can see here this expansion get increased here, and look at this area. You can see here the transformation. You can see the transformation here. Here you can see the microvilli, but here you cannot see the microvilli. It gets flattened, and also this area get widened. So this widening of expansion of this area is the result of the action of mucopolysaccharides. So these mucopolysaccharides are viscous compounds that absorb water to expand the space between this cell membrane and the fertilization envelope. Here you can see the substance released by the cortical granules absorb water and swell. And the third component of the inside the cortical granule is peroxidase enzyme and transglutaminase. And these two enzymes stabilize the fertilization envelope by cross-linking the adjacent proteins. And the fourth one is the hyalin that forms a coating around the egg. So hyalin forms a coating around the egg like this one, and which provides support for the blastomeres during cleavage. After the fertilization, the next stage is cleavage, isn't it? So during cleavage, this hyalin layer that formed during this slow block formation, the slow block process, provides the support for the blastomere during the process of cleavage. So you can read this description of this figure. We already explained this area, but you can read this description as well. So we can summarize the role of cortical granule proteins as this. The proteases digest the protein bridges that connect the vitellin envelope and the plasma membrane. Mucopolysaccharide produce osmotic gradient and water rushes into the perivitellin space, and peroxidases that hardens the fertilization envelope or provides the stability to the fertilization envelope, and hyalin form a hyalin layer around the fertilized egg and provides the support during cleavage. And this is the flow chart represents the complete action after the sperm egg fusion. This is the summarizing chart of egg activation after sperm entry. So the process of fast block and the slow block is also coming under the egg activation process, and but the egg activation process is not finished by the polyspermy prevention. We will see the further events in coming classes. Okay, hope you understood the topic well. Thank you.